Students, as we were talking about the disease in our last lesson, we are still continuing and we are today completing this topic. So, fighting disease. Medicine, the science of healing humans, was practiced in the ancient civilizations of Egypt, India, China, Greece, and Rome, and first developed by Islamic scholars in the Middle Ages. Gradually, medicine began to develop further in Europe and then eventually around the world. There are two aspects to dealing with this disease in medicine: prevention and cure. Prevention means not to be earlier, and cure when one of the persons infected or get the disease and to be treated from that disease. So, immunization is a major way of preventing some diseases, and the use of antibiotics has allowed many people to recover from disease who would otherwise have died. It means, guys, antibiotics, as they were investigated, they have huge vital importance for the health of the humanity. So the first antibiotic, how they were invented, let's say, the first antibiotic, bacteria and the fungi. They are, guys, we know that they are microorganisms, and they are the only microorganisms which can grow on a plates of agar. Agar, it's like jelly substance sealed inside of petri dish. Petri dish, it's this round plate where mostly the laboratory used in laboratory. Agar jelly contains the nutrients that bacteria and fungi need to grow and reproduce. It means once you put bacteria and the fungi to the petri dish which contain agar jelly, it means the bacteria and fungi will just grow. If you can find agar in the long way, I can give you one experiment. The experiment will be on the next page. There will be a link for the YouTube video, and you can do it at home to just check out how bacteria are all around of us. So, how it was first invented? There was a Scottish scientist called Alexander Fleming. In 1928, he was working in a laboratory when he noticed that the plate that had been set aside to grow colonies of certain bacteria also had a green fungus growing on it. There was a fungi and there was a bacteria also growing. It means at first it looked like as if the plate had been spoiled, but when Fleming looked again, he saw that the bacterial colonies near to where the fungus was growing had been destroyed. Here in the picture you can see the green one is fungus, and you can see most of the bacteria all around are destroyed. Grow. The reason that there was a substance in jelly which had come from the fungus, he thought like that killed the bacteria. But the fungus was called penicillium, which means little brush, like the brush, yeah. It was given this name because under the microscope part, the fungus looked like the real little brush. Here the picture you can see. When Fleming extracted the substance from the fungus, he called it penicillin. So nowadays, before nowadays, of course, Fleming tested penicillin on a range of bacteria and found that some were killed and others were not. He then tested penicillin on human white blood cells. You remember what is white blood cells? And found that they were not destroyed by concentrations that killed bacteria. So by this let's say experiment, Fleming understand that penicillin may be used in a cure way, but he didn't investigate penicillin. But the other scientists did, and from their work, a range of antibiotics has been developed to fight disease caused by bacteria in our daily life. It means many kind of antibiotics. For example, when we are when to be cured, contain small amount of penicillin, which are fungi, and those ones kill bacteria. So guys, we have finished the chapter like diet, drugs and disease. On our next lesson, even it will be offline or even it will be online, you're gonna have a small quiz. Be ready please for the quiz. See you soon.